Good day from Inside Drug Discovery. In this video, we will define drug discovery, distinguish between drug discovery and drug development, and list some activities that typically take place during the drug discovery stage. Let's begin with some characteristics of drug discovery. Drug discovery involves finding compounds with promising properties in laboratory tests and in animal studies. With regulatory approval, such compounds can be advanced into clinical trials for study in humans. Based on these characteristics, drug discovery is very often called preclinical research as well as non-clinical research. In short, the term drug discovery covers the non-human research required to create a drug intended for humans. Great, but what is the term for human-based drug research? Research that occurs in humans during clinical trials is called drug development. So while the terms drug discovery and drug development may seem synonymous, drug discovery and drug development are two distinct stages of drug research. While drug discovery strives to find and design compounds suitable for study in humans, drug development gathers evidence from human studies. These are clinical trials. Human study data, if they establish adequate safety and efficacy, may lead to market approval. We've now clarified the distinction between drug discovery and drug development. Occasionally, you might encounter the term pre-discovery. Pre-discovery covers activities such as identification of an unmet medical need, which is the opportunity for creating a new drug. With an unmet need selected, preliminary research on biological pathways might be required to understand how a molecular drug might affect a disease. The preliminary research normally identifies a specific target, usually an enzyme or receptor, that will be bound by the new drug. Finally, analytical methods for quantifying whether a compound is affecting the target will need to be refined. These analytical methods represent an assay for measuring compound activity. We're now ready to talk about early drug discovery. Most drug discovery programs start with using an assay to test or screen many compounds, perhaps even hundreds of thousands or over 1 million compounds, to find compounds called actives with promising target activity. Compounds that have confirmed activity upon retesting are called hits. In a process called lead selection, the hits are further tested to identify the most promising compounds, or leads. The best lead will be modified to improve its properties, improve its target activity, reduce its safety risks, and ensure the lead remains in the body long enough to exert its therapeutic effect. This process is called lead optimization. Early in lead optimization, the lead or its analogs will be tested with laboratory methods, in vitro tests. Compounds that show reasonable activity and safety in the in vitro assays will likely be advanced into animal studies, in vivo studies. The in vivo studies typically occur in the later stages of drug discovery. These studies gather evidence of in vivo efficacy and toxicology. Once a compound provides favorable in vivo results, all data on the compound will be submitted to a regulatory agency to enable the initiation of clinical trials. The US FDA process is called an IND, Investigational New Drug Application. The European Medicines Agency, EMA, calls this process a CTA, Clinical Trial Application. Drug discovery may continue with selected long-term toxicology studies, but the lead will advance into drug development and human studies. At this point, we have defined drug discovery, distinguished drug discovery from drug development, And name some of the research activities associated with drug discovery. Thank you so much for watching the video. Please like or subscribe or leave any comments below the video. Take care.